Principles of Inheritance and Variation, Session 2. In this module, we are going to learn about certain terminologies we come across while studying genetics. Gregor Johann Mendel is the father of genetics. He proposed several laws of inheritance by conducting extensive research and hybridization experiments on garden peas or Pisum sativum for about 7 years. Hybridization is the process of crossing two individuals differing in at least one character, resulting in a hybrid individual. Now let us learn why Mendel selected the pea plant. The pea plant is an annual plant, that is, it completes its life cycle within a single year. The flowers are bisexual, that means it bears a male and female sex organs on the same flower and they are naturally self pollinating. But yet, cross pollination can be conducted by emasculation techniques. The offsprings produced after cross pollinations are fertile, and pea plant has 7 pairs of contrasting characters which are visible to our naked eye. And the flowers of a pea plant are large enough for easy emasculation, and being a herbaceous plant, Mendel could grow large number of pea plants. Mendel for the first time applied statistical analysis and mathematical logic to problems in biology. He selected 14 pairs of true breeding lines, similar in all aspects, except for one character, to conduct cross-pollination or artificial pollination experiments. A true breeding line exhibits stable trait inheritance and expression for many generations due to continuous self-pollination. The 7 contrasting traits Mendel selected were the seed shape. Round and wrinkled were the contrasting traits of seed shape, seed color with yellow and green seeds, flower color with violet and white flowers, pod shape which were either inflated or constricted, and the pod color was either green or yellow. The contrasting traits for the character of flower position was terminal and axial, and for stem height, the contrasting traits were tall and dwarf. So these were the 7 contrasting traits that Mendel selected for his hybridization experiments. Use of true breeding lines helped Mendel to create a basic framework of rules relating to inheritance, which were later expanded as scientists discovered new informations pertaining to genetics. The data collected by Mendel was quite credible because he had used a large sampling size for his experiments. Moreover, as he conducted experiments on several generations of test plants, it helped to confirm his inferences and prove that his laws were not unconfirmed ideas but general rules of inheritance in all living organisms. However, the world had to wait till the end of 20th century to understand the significance of Mendel's discovery. Due to his valuable research in the field of genetics, Mendel came to be known as the father of genetics, and the rediscovery of his laws laid the foundation for modern science of genetics. Now let us learn a few terms we come across while studying genetics. Gene or factor is a functional unit of a hereditary material, that is DNA. In other words, it is the unit of DNA that is responsible for the appearance or inheritance of a character in a living organism. The gene is a functional unit of a hereditary material. The alternative form of a gene located at a specific location on a specific chromosome is known as an allele. It governs a trait of an individual and is generally represented by a letter. Trait can be defined as a distinct variant of an original phenotypic character, which may be inherited or environmentally determined. For example, color of a flower or petal is a character, while the colors pink, violet, and white are traits. The genetic makeup of an organism is called as a genotype, while the appearance of an organism is given by the term phenotype. Every organism has two alleles for each character. Every organism has two alleles for each character. If these two alleles for a particular character is identical, the gene is said to be homozygous or pure, and the individual is called a homozygote. On the other hand, if the individual or an organism possesses two contrasting alleles in a pair, the gene is said to be heterozygous and the individual is called heterozygote. In a heterozygote, only one of the contrasting genes is able to express itself, while the other gene remains hidden. The gene which expresses in the F1 hybrid is called as the dominant gene, while the gene which is unable to express itself in the F1 hybrid is called the recessive gene. F1 hybrid of first filial generation is the first generation of plants or animals obtained by crossing different parent types. When two individuals of the first filial generation are self-fertilized, the resulting progeny is called the second filial generation or F2 hybrid. Self-fertilization is a term used when fertilization takes place between the male and female gametes from the same individual. The study of inheritance of one pair of contrasting character is known as a monohybrid cross. For example, the study of inheritance between tall and dwarf plant. The study of inheritance of two pairs of contrasting characters is known as a dihybrid cross. For example, the study of inheritance of round and yellow seeds with a wrinkled and green seed is an inheritance of two pairs of contrasting character. To determine if the individual is homozygous dominant or heterozygous recessive, a backcross is carried out. A backcross is a cross between the F1 hybrid generation and any of its parents, whereas a test cross is a cross between the F1 generation progeny with a homozygous recessive parent. It is done to know an unknown genotype. Thus, genetics helps to understand how traits are passed from parents to offsprings, and the principles governing inheritance was discovered by Gregor Johann Mendel, the father of genetics. Dear children, in this module, we learnt about certain terms we come across while studying genetics. Thank you.